Hello, my name is Siobhan Green. It is week 5 of microbiology. This week we are working on biochemical testing for microbial identification, carbohydrate fermentation testing. Right now I am letting my tubes, I just took them out the refrigerator. It says to let them get to room temperature approximately about 30 minutes. But as they're warming up, I'm going to go ahead and do the first step, which is to take my permanent marker and label my 6 phenol red vials. So I'm going to go ahead and begin doing that. So the first one would be my S cerevisiae glucose. So I'm going to go ahead and mark that one as my number 1 tube. Okay, so I have that one labeled there. And I'm going to go ahead and finish out with the other 5. Okay, so my next step is to place my 6 Durham tubes and my 2 dropper pipettes into my cup of alcohol. So I'm going to go ahead and fill that cup of alcohol. And place my pipettes in there. Make sure I have 6 of these here. Yes, I do. So I'm going to go ahead. Oh no. Go ahead and place these into my cup of alcohol as well. Okay, so my next step is to remove the cap off of my first tube. Sterilize it. Carefully transfer half of the glucose into it. Sterilize it again. Cap it. Take my second one, which is my S. epidermidis. Sterilize it. Take my remaining glucose and insert it in. Sterilize it. And put the cap back on. And I'm going to do that with my 4 remaining tubes. However, it won't be glucose. 2 of them will be mannitol and 2 of them will be fructose. So my next step is to remove my Durham tubes and allow them to dry thoroughly. Okay, I'm going to go ahead and let those dry. So it says to insert 1 Durham tube into each fennel broth and making sure that the opening of the Durham tube is going downwards. So I'm going to go ahead and proceed with putting the other 5 in. Okay, so now I am opening up my active broth of my S. cerevisiae. I am sterilizing that. Taking my pipette, shaking it dry, And in each of my S-Cerevisiae tubes, I'm putting 4 drops in. I am going to re-sterilize my active broth, cap it, and I'm going to proceed to do the same thing with my S. epidermidis with my other 3 vials. Okay, so now that I'm done, I'm going to put these in my tray here, and I'm going to let them incubate for 12 hours. And I will come back and check on those and be back with the video. Thank you. So it's been about 15 hours since I've incubated my red fennel broth. It says now to basically just gather my fennel red broth and note the color of the solution, if there's any gas bubbles in the little Durham tubes that we inserted into the vials. And it wants us to categorize. I kind of separated mine, so I have 3 of them that kind of still have that little yellowish, and this one's still kind of red fully, but these 2 are kind of like the yellowish to reddish changing. And the 3 that actually went full yellow for me was my, my S. cerevisiae. They're all S. cerevisiaes for glucose, fructose, and my mannitol. So, and the only one that really I see has gas bubbles is my fructose one. So I would say this one is probably my fermenter with acid and gas production, just because those gas bubbles are in there. These 2 I know are fermenter also. I don't see any gas bubbles in them. This is my S. cerevisiae, my mannitol, my glucose. There's at the top a couple bubbles, so I would say all my S. cerevisiae are maybe fermenter with acid and gas production. My S. epidermidis, fructose, and mannitol have changed, but they're kind of still like that reddish color. So I believe that these are both fermenter with acid production only. And then this one, my glucose, is still red. That's the only one that's really still red that didn't really change colors. So I would say that this one, which is my S. epidermidis glucose, is non-fermenter. So now the directions just tell me to go ahead and record all my observations in my lab assistant, and then I will take pictures and insert those also. And our carbohydrate fermentation testing is finished. Hope you enjoyed the video. Thank you.